The gram staining technique was first introduced by a Danish bacteriologist named Hans Christian Gram in 1884, and this was the basic primary or preliminary screening technique that has been used in the microbiological laboratory since it was discovered. It was from 1884 to till now we are using the gram staining method. So there is no change in that. Nothing is changed. So the gram staining technique was discovered by the Hans Christian Gram. He was a Danish bacteriologist in 1884. Till now, most of the laboratory, research laboratory, hospital, clinical laboratories, microbiological laboratories, research, everywhere we are using the gram staining is the basic and preliminary technique. So it is widely used method to differentiate and classify bacteria on the basis of their staining reaction. So it is a widely used method to differentiate and classify bacteria on the basis of their staining reaction. So this method is used to differentiate or differentiate the bacteria based on their staining reaction. Why? Because the bacteria, so there are more than 13,000 species of bacteria are there. More than 13,000 species of bacteria are there. So as bacteria are colorless, they are colorless. So we cannot identify, we cannot differentiate the bacterial species. So for that, we need a staining technique. Staining means nothing but a color dyeing technique. By utilizing this technique, we can classify the bacteria into two categories, either gram positive or gram negative, like that. So what are the materials required? Just inoculation loop we want. You know that inoculation loop. What is the special in that inoculation loop? So this is the holder actually, and this is the holder, and this is the loop portion. So this loop actually it is like a wire. It was like a wire, but this wire is made up of, instead of, if you just find in a normal electrical wire, so that will be insulated. The wire is insulated with a plastic or PVC, like that. Inside that there will be either aluminum or copper will be there. But here this is made up of nichrome. That is called a nichrome. This is made up of nichrome. The speciality of the nichrome is it will immediately get heat when it is introduced into the heat. It will immediately get the fire, gets catch fire, and becomes red hot. And then within 30 seconds, it will once again cool. If you take aluminum or copper or whatever the metal, once it gets heated, once it becomes red heat, then it will not be cooled suddenly. It will take half an hour, half an hour, 1 hour, like that. But here in this case, as it is made up of nichrome, once it get heated, and at the same time immediately it will get cooled, it will be cooled. And you know that this is a microscope, this is a compound or bright field microscope, whatever, binocular microscope, whatever you can say, right? You know that microscope. Then you need, this is the primary stain, crystal violet. This is the primary stain, crystal violet. Then there is a modern grams iodine, decolorizer, acetone or ethyl alcohol, and then counter stain, either dilute carbolfuction or safranin. Dilute carbolfuction or safranin we can use as a secondary stain or counter stain. So the primary stain is the crystal violet, then grams iodine is the modern, then decolorizer is the acetone or ethyl alcohol, and counter stain dilute carbolfuction or safranin. Then as bacterial culture, so the bacterial culture should be like smeared like this one, bacterial culture. Then apart from this, you have to take either you should split lamp and glass lamp. So these are all the requirements for the practicals. So coming to the procedure, so if you want, I will send you the recording also. If you want, I will send you the recording also. I will just record and I will send you the file also. So coming to the procedure, you just take a bacterial culture or bacterial suspension bacterial culture, just smear in a clean glass slide. First, we have to make a clean bacterial smear, a clean glass slide, and just next heat fix. You know what is heat fixation? You just after smearing, after the bacterial smear is made, then you just gently expose the slide in front of the flame. Just gently expose in front of the flame so that the bacterial suspension will get dried and it will be fixed in the slide. It will be fixed in the slide. You should not heat until for a long time. Why? Because if you heat, then the cell wall will be broke down. The bacterial cell wall will broke down, and even the glass slide will also broke down. Even the glass slide also getting breakdown. So you just gently expose the slide into the heat so that the moisture content in the suspension will evaporated and the bacterial culture will be dried and fixed in this slide. Then after that, you just prepare a tray, staining tray. You just prepare a staining tray. In that, you just keep the slide and add a few drops of crystal violet. That is the primary stain. You just add few drops of crystal violet just in the slide. Then you just wait for 1 minute for the reaction so that the crystal violet will be allowed to react for 1 minute. So you have to wait for 1 minute so that it will get reacted. Once the crystal violet is added, it stains all the bacteria as purple color. Why? Because the crystal violet is purple in color. So once it is added, then all the bacteria will be purple in color. Why? Because it is the primary stain. Once it is added, then it will become purple color. Then after that, then after that, the smear was washed with running tape water and dried. Smear, we have to wash the tape water, wash the smear with running tape water, and we can dry. Just you have to dry. Then, then the next step is you have to add grams iodine, a few drops of grams iodine. The role of grams iodine is nothing but it acts as a modern so that it will increase the affinity. It will increase the affinity of the crystal violet to the cell so that the cell will become highly attached to the primary state. So it will increase the affinity attraction. And the grams iodine is added and allowed to react for 1 minute. Then the smear is once again decolored, means you just add a few drops of acetone or alcohol drop by drop. You just add acetone or alcohol drop by drop so that until the washing solution contain no crystal. It just once you add, then it will wash out the excess crystal oil. It will wash away the excess crystal oil. So the wasting solution will be violet in color. So this will be violet in color. You have to add up to, you have to wash the slide up to no blue color is in the solution. So that the excess staining will be removed from the slide. The excess staining will be removed from the slide. Then once again, you have to wash the slide in the running tape water. You just wash the smear in the running tape water. Just once again, you wash the smear in the running tape water. Then counter stain with dilute carbol factor. Then you have to add the counter stain or secondary stain. The counter stain is also called as secondary stain. You have to add the counter stain or secondary stain, dilute carboxyl or safranin. You can add dilute carboxyl or safranin. It was poured over the smear and it should be allowed for 30 seconds to 1 minute. Just wait for 30 seconds to 1 minute so that uh, it will react. Then after that, the smear was washed in running tap water. You can wash the smear in running tap water and you have to make it dry. So what happens? Then the slide was dried and observed under microscope. The slide was dried and observed under microscope. Then after that, you can see after completion of the 4 steps, that is primary addition of primary stain, addition of moderate grams iodine, then addition of acetone decolorizer, and the addition of the secondary stain, safranin or carboxyl. If the organism just retains the primary stain crystal violet, then they are gram positive. Why? Because some bacteria, once it is fixed with that, once it is fixed with the crystal violet, then it won't take the other staining solution, that is the carboxyl or saffron. It won't take. It will not take. Once why? Because the cell wall will be fixed. The cell wall will be attracted and stained with the crystal violet. So even though decolors are added, even though the moderants are added, even though the secondary stain are added, it will not be fixed. If they get some bacteria are there, some slides are there, some bacteria are there. In the initial stage, it will absorb the primary stain, crystal violet. In the initial stage, while we are adding the crystal violet, it will absorb, it will retain, it will absorb the what do you call the crystal violet. But while you are adding the acetone or ethyl alcohol as the decolorizer, that it will loses off. That means it will loses off the stain. And then once again, what is the next stain we are adding? We are adding the secondary stain or counter stain, that is the safranin or the carboxyl. And then it will take this safranin or the carboxyl and they will appear as pink or red in color. And they will appear as pink or red in color. So based on their staining reaction, almost all the bacteria can be grouped into can. So based on this reaction, we can group all the bacteria into either gram positive or gram negative. So all the 13,000 species of bacteria can be grouped under either gram positive or gram negative, right? So what are all the bacteria which are discovered so far, we can group them into either gram positive or gram negative. So after the steps, if it is gram positive, the bacterial cell wall, the bacterial culture, bacterial cell will be appear like this. So this is the purple in color, you know. So after all the steps, you can see if it is gram positive, we can find this type of colony, these type of cells. The cell will be purple in color. Actually, this is a staphylococcus. This is staphylococcus. It is a gram positive cocci, you know very well. The staphylococcus is a gram positive cocci, you know very well. It will be like this. Now, if it is gram negative, then it will be like pink color. Because it will, even though it utilizes, even though it absorbs the crystal violet, but when we are adding the decolorizer acetone or ethyl alcohol, it will loses the crystal violet. And after that, when the safranin or the carboxyl is added, it will absorb with the safranin or the crystal carboxyl, and it will be pink in color. Why such some bacteria utilizes the primary stain crystal violet and some bacteria utilizes the secondary stain safranin, in the sense that is due to the presence of the amount of peptidoglycan in the cell wall. So that is due to the presence of amount of peptidoglycan in the cell wall. Some bacteria possess 20 to 80 nanometer thickness of peptidoglycan in their cell wall. Some of them will contain only 20 to 80 nanometer thickness of the, 2 to 8 nanometer thickness of the cell wall. Some bacteria contains 20 to 80 nanometer thickness of peptidoglycan, that is a polysaccharide that will be present in the cell wall of bacteria, whereas very few bacteria contains only just 2 to 8 nanometer of peptidoglycan in their cell wall. So that is the reason. So once the peptidoglycan is highly present, that is 20 to 80 nanometer, it will absorb the crystal violet and it won't loses the crystal violet. But as the some bacteria contains only fewer amount of, that is 2 to 8 nanometer of the peptidoglycan, even though it utilizes the crystal violet, but when we are adding the decolorized acetone or alcohol, it will loses the crystal violet, and when we are adding the carbon function or saffron, it will absorbs the secondary stain and it will be pink in color. So that's all about the gram staining procedure. And then up to this, we can first thing I will show you in the video in the next class. I think we can complete the practical now, and we'll discuss the practical in the next Saturday. Okay. So in May hereafter every Saturday we'll be having practical. So I will mark the attendance for both the session, or because we have crossed the both the section. So I will mark the attendance. I think it is, what's the time now? 3:02 only. 45. So I will mark the attendance for both the session. So you'll be free now. And from next Saturday onwards, we'll do the practical. I will show you somewhat video, and there are a lot of video I can personally do did it during my laboratory. I will share the videos also. Okay. If you want the recorded session or PPT, I will share you in the group also. If you want the PPT, you just message me either personally or in the group. I will send the PPTs, that is PPT, to your group also. Okay.
Okay, sir.
Okay. Bye. Take care, all of you.
Thank you so much, sir. Bye.
Happy,
Take care, sir.
happy
Bye,
weekend.
sir. Take care. Good night. Oh,
Good
sorry.
night. Happy,
नहीं क्या था? Good morning. नहीं, good afternoon.
oh, happy weekend. Happy weekend.
Thank you.